Welcome to the Vault, Director. As requested, we've prepared the Arklay incident report for you. It entails an overview of every organism encountered in the outbreak at the Umbrella Laboratory, as well as a comprehensive analysis of their biology. Let me walk you through them one by one. Specimen 001, a former staff member who was unwillingly exposed to the experimental T-virus during the containment breach at the Arklay facility. The early researchers who had been working on the strain gave a crude designation to its victims: zombies. The name endures not for scientific accuracy, but for its grotesque truth, for they are in essence the dead forced back into motion. The T-virus is a recombinant pathogen designed by our laboratory as a military bioweapon. Its mechanisms combine rapid cellular necrosis with equally fast cellular regeneration. Whenever a human host is infected, the following sequence occurs. Stage 1. Exposure and transmission. This is where a primary vector contaminates the subject. Infection includes bites, scratches, and even contamination through mucous membranes, such as infected blood or saliva. Secondary exposure occurs via aerosol particles in high concentration, usually in incidents such as the one we experienced in the Arklay incident. Stage 2 is the incubation period. Here symptoms will manifest within hours, where the initial signs include fever, chills, nausea, and violent migraines as the virus penetrates the nervous system of the subject. What soon follows is a lapse into a coma-like state, as well as systematic failures of most organs within the body. What follows is then stage 3, the neurological hijacking. Here the virus targets the brainstem, destroying any higher cortical function, while stimulating more primitive regions, such as aggression, motor reflexes, and most importantly, the hunger impulses. At this stage of the infection, the prefrontal cortex begins to decay, eliminating memory, morality, and reasoning, and what remains now is only instinct. It is in this stage where the victim ultimately succumbs to the virus. This leads us to the fourth and final stage of the infection: reanimation. Despite organ necrosis, the regenerative properties of the virus keeps tissue viable. Muscle fibers begin to stiffen, resulting in their clumsy erratic movement. The cardiovascular system fails, leaving the body cold, pale, and in various states of decomposition. This is for the majority of subjects infected, the final stage of the T-virus, and they will remain at this stage until the instability of the T-virus finally destroys the subject completely. As per policy, I have included a tactical assessment for the company as well. Individually, these organisms, designated as zombies, are slow and limited in motor coordination. However, their resilience to pain and injury makes them quite difficult to neutralize. There is also the potential risk of infection from even a minor bite or scratch, and once a victim has been contaminated, there is no cure. Their numbers multiply exponentially in confined environments, which all graphs and analysis indicates could be catastrophic if the correct field conditions are met. As a closing note, the outbreak in the Arklay facility has marked the first true demonstration of the T-virus's potential, as well as its uncontrollability. And while many of my colleagues are quick to deem the zombie both a failure in terms of science and its weaponization, I think that the properties contained within this strain could be absolutely devastating if deployed correctly. Specimen 002, the Cerberus. This is another part of our animal testing program, specifically bred Doberman canines exposed to the T-virus in controlled laboratory conditions. The designation Cerberus was assigned internally by Umbrella staff, as they hope to classify these creatures as official bioorganic weapons. Yet it is important to note the distinction between the Cerberus and ordinary infected canines. These dogs were deliberate test subjects, whereas other dogs, particularly those seen later during the Raccoon City incident, were simply stray animals infected incidentally by coming into contact with the T-virus strain outside of the laboratory. These stray dogs will be further cataloged in the Raccoon City files, as they are viewed as a separate strain compared to the engineered Cerberus. The Cerberus program was designed to observe the viral impact on highly trainable, fast-moving animals that could serve as attack units, potentially in war zones. These infected canines display the following progression. Stage 1. Exposure and transmission. Here the canines were subjected to laboratory inoculation through direct injection of the T-virus strain. Stage 2 is the incubation period, where symptoms appear within hours. However, unlike the human subjects, canines do not lapse into comatose states. Instead, much like an infection of rabies, the subjects grow increasingly aggressive and restless as viral penetration accelerates through their nervous system. Stage 3 is the neurological effects, where the destruction of higher brain function is rapid. Any obedience training, no matter how disciplined the dog is lost within a day. Instead, the virus reinforces primitive hunting and pack behavior instincts, allowing the Cerberus to conduct highly coordinated group attacks, despite their severe mental degradation. Stage 4 is the physiological mutation, where the severe necrosis of dermal tissue results in exposed musculature and bone, giving the infected dogs their grotesque and skinless appearance. The enhanced adrenal response by the virus allows for short bursts of speeds far greater than any baseline dopamine, yet the respiratory instability and organ failure leads to eventual total collapse, though most of the subjects remain highly lethal long before this shutdown happens. Yet despite the initial promise of the Cerberus project, we have observed several side effects and limitations that drastically impact the overall performance. Their durability is vastly lower compared to larger bioweapons. Their metabolic instability also causes a rapid deterioration of the subject. This means that Cerberus units are much shorter lived compared to their human zombie counterparts. On top of that, the viral overactivity within their bodies leaves them prone to collapsing under stress, and this instability was noted to be the biggest flaw within their first deployment. From a tactical perspective, the Cerberus are significantly more dangerous than baseline zombies. Their speed, coordination, and unpredictability make them the ideal shock troops in confined environments. But besides that, their psychological impact on targets is considerable as well. The sight of once loyal, domesticated animals has been documented to often cause hesitation in untrained personnel. However, their deterioration, as well as their biological instability, leaves much room for improvement, and I would highly recommend refinement of this particular program before we move on to the stages of field testing. Specimen 003, the Crow. This is but a scavenger bird found within the Arklay region, which has been infected through contact with contaminated tissue from other experimental organisms. Unlike the humans or canines, these avian hosts were not deliberately exposed in laboratory conditions to the T-virus. Their infection was incidental, born from the Arklay lab staff members' inability to contain the outbreak. Yet while their presence is a byproduct of a failed security protocol, their presence is interesting as it illustrates the T-virus's dangerous adaptability. Through this report, we have documented that even species not targeted for research can become vectors if exposed to the strain. Once infected, these crows adapt a far more aggressive temperament, demonstrating just how easily this virus can weaponize natural behavior. Yet the pathogenesis of the T-virus in avian hosts is different to that within the mammalian system. This results in less pronounced physical mutation, but instead reflects itself through extreme behavioral changes. Stage 1 of their infection is exposure and transmission. Here the crows will have ingested either contaminated tissue or contaminated waste from around the mansion grounds, and it seems that this ingestion is more than sufficient enough to pass on the virus to the animal. What follows is Stage 2, the incubation period. Just like both humans and canines, the symptoms manifest rapidly. Yet unlike humans, the birds rarely exhibit the fevered collapse, and instead the host will remain active, while the virus slowly rewrites their neural chemistry. What follows is Stage 3, the neurological effects. Here the T-virus heightens aggression and reduces flight inhibition. Seen typically as skittish scavengers, the crows suddenly begin to exhibit persistent hostility towards anyone in their proximity. Yet despite their infection and lapse in cognitive function, it seems that they, much like the canines, retain the ability to work together in large numbers and will both hunt and attack in large swarms. What follows in stage 4 is the physiological mutations, and unlike humans and canines, there is no extreme size increase nor necrosis observed. The mutation on the birds is subtle, confined largely to heightened resilience and endurance. Important is it to note that the primary danger of the crow lies not in its appearance, but in their numbers and behavior. In a tactical assessment, though the crows are fragile compared to other infected hosts, their role as a secondary vector cannot be overlooked, for each contaminated bird carries more than enough contaminated fluids to be able to spread the infection further. It is because of this that me and my colleagues have deemed that the fragile bodies of the crows, though lacking the immediate lethality to be considered a true bioorganic weapon, has a much greater potential significance when it comes to spreading the immediate infection of the virus. Their swarming behavior and high aggression makes them particularly dangerous to unprotected personnel, especially in confined outdoor areas. And while individually fragile, in great numbers they can serve as an immediate catalyst for deploying any type of infection, and should containment of that initial outbreak fail, then I see limitless possibilities in terms of spreading other pathogens in war zones. Specimen 004. This is Hunter Alpha. Unlike the zombies, the Cerberus, or the infected crows, this creature is not the result of random infection for the T-virus. The Hunter Alpha represents one of the Arklay facility's most deliberate and ambitious creations, a true bioorganic weapon, engineered from the ground up. The Hunter project began with human embryos, genetically altered and fused with reptilian DNA. The T-virus was then applied to stimulate growth, harden physiology, and strip away any cognitive restraint. The result was a humanoid predator that retained enough intelligence to follow orders, but also enough aggression to ruthlessly chase down prey. Unlike the mindless zombie, the Hunter Alpha was designed to stalk, strike, and kill with precision. Once matured, these subjects grew to nearly 2 meters in height, their musculature heavily defined, and their bodies sheathed in a leathery reptilian skin. Their elongated claws were not mutations born of accident, but intentional weapons, natural blades capable of tearing through steel mesh and body armor alike. This particular subject caught our attention as they, among our earlier projects, were completely unmatched in terms of speed and agility. Where the zombies staggered in sluggish persistence, stiffened by the decay of necrosis, the hunters were not only able to do full-speed sprints, they were also capable of leaping upon prey and decapitating them with a single strike. Neurologically, the Hunter Alpha has retained fragments of the human brain structure, but only what our researchers at Arklay deemed useful. The cognitive functions was reduced to the bare minimum: recognition of commands, basic pack coordination, and even limited problem solving in the pursuit of prey, such as the capability to walk through doors. What remained within the human test subject was consumed by reptilian aggression, possessing the intelligence of an orangutan, as well as a cold and unrelenting drive to hunt anything that moved. Unlike zombies, Cerberus, or even the crows, the incubation process for a hunter was not measured in hours or days, but rather in months of accelerated growth, where most hunters would reach maturity after a year in development. The documentation from the Arklay incident shows us that while zombies could be somewhat avoided, distracted, or even outpaced by running, the hunters, on the other hand, were relentless and offered no such mercy. Their agility allowed them to scale walls, leap across distances, and their heightened senses made stealth nearly impossible. Every documented encounter with these creatures became not a contest of endurance, but rather a matter of survival, measured in mere seconds. Yet the Hunter Alpha was not without its flaws. Controlling the creature became a tenuous process, for while capable of recognizing its handlers, the risk of them turning on other Umbrella operatives remained high, with lethal consequences every time. Yet the tactical significance of the Hunter Alpha cannot be overstated, for it was one of the first research projects within the Arklay facility that truly proved that our ambitions were not limited to viral accidents, and a conscious step toward our ultimate goal of creating the ultimate biological soldier. This is Specimen 005. The Chimera represents one of our most grotesque and failed experiments. It is a project that sought to splice the human genetic material with that of flies, and then force the resulting embryo to maturity under the influence of the T-virus. Where the Hunter Alpha was engineered as a weapon of deliberate precision, the Chimera was an abomination born from recklessness, what we deem a genetic crime that merged both man and insect into something that neither could recognize. Even from the earliest stages, the Chimera subjects were designed not to resemble soldiers, but instead testbeds for hybridization. The result was a twisted parody of a human form, elongated limbs, patchy exoskeletal growth, and a body riddled with deformities. Their skin peeled away to reveal chitin-like plates across the torso, while their heads retained only the vaguest of human outlines, mutilated by compound eyes and grotesque mandibles. The infection process was unlike that of ordinary humans, for the T-virus did not simply reanimate the dead, it accelerated insect DNA spliced into the host genome, forcing radical mutations that rendered the Chimera unstable from its very conception. Many of the test subjects perished during gestation, and those that survived emerged as malformed creatures, barely able to walk, their erratic movements betraying their own bodies, constantly in rebellion against themselves. Behaviorally, chimeras exhibited both human and insect traits. They scuttled along walls and ceilings with unnatural agility, striking victims from above with their hooked claws. Their reproductive systems had been warped by the viral cocktail, producing massive larval sacs that clung to their deformed abdomens. There are even reports that indicate they attempted to implant these larvae into humans, essentially a grotesque mimicry of their parasitic behavior. The project was a failure through and through, as physically, their endurance was insignificant compared to hunters. Their bodies, ravaged by instability, were fragile, and their movements was erratic and unpredictable. The only positive outcome from a tactical perspective was that they posed a decent threat in swarms, as they could overwhelm victims not with durability, but through sheer terror and numbers. However, it is speculated that this minor efficacy was contributed to the psychological effect of encountering such a creature, as they in terms of physicality and durability posed minor threats to military personnel. Ultimately, the project was deemed a failure. Their instability made them impossible to mass produce, and their grotesque appearance undermined their potential for sale on the black market, and thereby field deployment. We within the Vault have deemed them too monstrous, too uncontrollable, and too fragile to serve as practical weapons. This is Specimen 006. The project was named Neptune. Unlike the incidental infections of the crows or adders, this organism was a deliberate test case. Our researchers exposed a captured great white shark to the T-virus in hopes of creating a powerful aquatic bioweapon. Initially, at first glance, the results seemed promising. The host grew larger than expected, its musculature thickened, and its aggressive behavior intensified. Yet the researchers soon realized that the T-virus had done little to truly improve the shark beyond its natural state. For after all, a great white shark is already, and has been for millions of years, one of the most efficient apex predators on the planet. The virus merely amplified its aggression, but offered few new traits or abilities to its already lethal capacity. Worse yet, and probably the biggest Achilles heel of the project, was that the Neptune remained entirely bound to its aquatic environment. Out of water, the creature was essentially helpless. These limitations marked the project as a disappointment, for where the zombies proved able to spread the infection by overwhelming subjects with their resistance to physical trauma, and the Hunters project broke all expectations with their strength, agility, and ferocity, the Neptune experiment had nowhere to go, except for the confinement to water. Because of that, its reliance on containment tanks within the mansion laboratories made it more a liability than an asset. Ultimately, what we in Umbrella sought was the perfect soldier, mobile, versatile, controllable, yet Project Neptune was none of those things. Though to clarify from a tactical standpoint, disappointment does not mean harmless. Within the laboratory, the infected sharks, already apex predators, were driven into violent frenzies by the viral overstimulation. Their attacks had become faster, their behavior more aggressive, and they would now pursue victims relentlessly compared to unaltered sharks. In the claustrophobic tanks beneath the Arklay laboratory, they were more than capable of killing anyone unfortunate enough to fall into their domain. There are even reports that suggest that even specimens weakened by the T-virus's instability as well as age and environmental stress could still capsize entire containment platform and batter reinforced steel barriers. What we ultimately learned from the Neptune's creation was not triumph, but that even the T-virus has its limitations. Essentially, the strain can warp and weaponize basically any deadly hunter, but that does not mean that all results are practical. For in the end, a weapon without versatility is not a weapon at all. This is Specimen 007, a spider enlarged and weaponized by exposure to the T-virus. Unlike the accidental infections of crows or snakes, these creatures were deliberately bred and observed within the Arklay laboratories. Our researchers have given them the official codename, Web Spinner. The choice of spiders was not random. Our researchers selected them for their natural hunting abilities, their inclination to stealth, climbing, as well as their venom. When exposed to the T-virus, their physiology responded with dramatic results. Subjects grew to several meters in length, their musculature and exoskeleton reinforced to carry the weight of their now swollen frames. The transformation was both grotesque and unstable, leaving them lumbering, yet powerful predators. The infection process amplified their most primal of traits. Their venom glands grew enlarged and produced copious amount of toxic saliva, powerful enough to corrode flesh on mere contact. Their spinnerets adapted to expel unusually strong silk, allowing them to coat entire corridors and ensnare prey with alarming efficiency, yet their aggression remained largely instinct-driven, far more animalistic and unpredictable compared to the control, engineered-like precision of the Hunter Alpha. Within the Spencer mansion, the web spinners demonstrated dangerous adaptability. They clung to ceilings, dropped from shadows, and spat venom to weaken victims before closing in. Even gunfire often failed to stop them immediately, as their thickened carapace allowed them to absorb immense amounts of damage that would undoubtedly have killed any ordinary arachnid instantly. Their physiology, however, carried the incessant fatal flaw of the T-virus, internal instability. Yet one interesting fact about the web spinners is that their carcass would often rupture upon death, releasing smaller parasitic offspring. These spiderlings were much weaker, but still venomous, perpetuating the lethality of the web spinner, even beyond its death. Behavioral observation noted that despite their size, the web spinners did not lose their territorial instincts. They congregated near nests and web-coated chambers, turning entire rooms into absolute death traps. Their mobility and ability to operate in both open and confined spaces made them more than simple test animals. For our researchers and for Umbrella as a whole, they were proof to us that the T-virus could indeed create predators out of even the smallest of subjects, showcasing the limitless potential within this miraculous strain of infection. Yet similar to the Neptune project, the web spinners carried inherent limitations. Their size made them impossible to conceal, and their dependence on instinct made them uncontrollable. They were more a spectacle of horror than a weapon of war, and in the controlled corridors of the Arklay facility, they might have served their purpose as deterrents, but as battlefield assets, we have deemed them to be failures. This is Specimen 008, a small snake native to the Arklay region, unintentionally infected through exposure to contaminated corpses, as well as laboratory waste. These creatures are thereby not selected as deliberate test subjects, yet just like the crows, they have piqued our curiosity as they became accidental vectors after the outbreak. Their codename within the Arklay Files has been labeled as Adder. Unlike the colossal Yawn, the adders did not grow into towering predators from exposure to the T-virus. Instead, the strain produced subtle yet effective changes within their biology. Just as anyone else exposed to the virus, the subjects displayed increased aggression, heightened resilience, as well as a similar tendency shown in the crows to congregate in swarms, further emphasizing that somewhere within the genetic strain of the T-virus exists the capacity for improving eusocial behavior. Though individually fragile, their danger lay in numbers, as the adder will strike repeatedly from corners, cracks, and shadows. The infection of the T-virus also altered their venom, amplifying its toxicity. Even small bites have shown to deliver heavy doses of neurotoxin, capable of overwhelming the human nervous system with ease. Patients exposed to the venom has described symptoms of dizziness, blurred vision, and even progressive paralysis following even a single bite. Yet unlike the zombies, which spread the infection through their saliva, the adders propagate terror through venom alone. It seemed that their role in the outbreak was deemed to be less about transmitting the virus and more about creating chaos, exhaustion, and attrition among people exposed to the environment around the virus. Similar to other animals, their incubation was swift. Once exposed to contaminated material, adders displayed aggressive behavioral changes within mere days. Their physiology remained largely recognizable as snakes, though signs of viral instability were visible, indications such as mottled scales, pallid coloration, and sluggish movement offset by sudden bursts of striking speed. Inside the mansion and its surrounding grounds, much like the crows, they became an environmental hazard, found in basement corridors, ventilation shafts, and overgrown areas. The adders thrived in confined environments. The greatest danger was not their size nor their strength, but rather their ability to overwhelm victims with multiple simultaneous strikes. From a tactical perspective, the adders are of little interest to us as a company. They lack the capacity of the crows, the controlled lethality of the hunters, or even the sheer resilience of the simple zombie. Yet their presence, combined with the existence of the infected crows, reveals another fascinating truth about the T-virus. Once released, the virus is a catalyst for contamination, discriminating against nothing of biological access, infecting not only the mighty, but the insignificant as well. Specimen 009, a giant snake residing within the Spencer mansion, transformed by the mutagenic effects of the T-virus into a predator of impossible scale, designated Yawn by our researchers for its cavernous gaping maw. This organism represents the full extent of viral overgrowth if applied to a reptilian host. Unlike the smaller adders, which carried the virus incidentally, Yawn was a deliberate test subject, a native serpent captured for study, which through the trials responded violently to its infection. Rapid and uncontrolled cell growth produced its immense size, easily dwarfing its original form. The result was a creature more than capable of swallowing a grown man whole, another perfect expression of the T-virus's capacity to warp natural predators into enormous monstrosities. Yet the infection did not simply increase Yawn's size, its musculature was grotesquely strengthened as well, giving it the power to smash through walls and coil itself together with bone-crushing force. Viral amplification of its venom gland resulted in a potent neurotoxin far more dangerous than any natural snakebite. Survivors of its attacks have reported progressive paralysis, seizures, and eventually death if left untreated. Field studies have shown that even minor exposure to the venom has weakened both mental and motor functions, making escape after physical contact almost impossible. Physiologically, Yawn retained the basic structure of a serpent, but exhibited grotesque mutations along the jawline and spine. Its fangs, lengthened and reinforced, were capable of piercing reinforced materials, while its widened jaw could easily engulf a prey with a single strike. In terms of behavior, Yawn also displayed more intelligence than that of the adders. It was territorial, capable of ambush, and sensitive to vibrations within its environment. Unlike zombies or the Cerberus, which would both attack mindlessly, Yawn displayed a predatory patience, lying in wait for its victim before striking with deadly precision. Yet despite its size and power, we have ultimately labeled Yawn a disappointment within the research department, for its size renders it not just uncontrollable, it also makes deployment a nightmare, if not completely impossible, which means that just like the Neptune project, it is impressive in isolation, but impractical in its usage. Once again, the T-virus had created a monster, not a soldier, and while it within the sealed walls of the Arklay facility was more than enough to inflict casualties, there is simply not enough evidence to suggest that moving forward with this project will garner any success down the line. Specimen 010, also known as Plant 42, is one of the more interesting entries in the Arklay Files. What began as an act of scientific curiosity rapidly transformed into one of the most grotesque anomalies in the entirety of our company's history, as it is a stationary organism that has, through the T-virus, learned to kill. Through unknown factors we have yet to map, Dr. Henry Sarton unlocked the potential within the plant that goes far beyond its natural limits. What type of plant Dr. Henry has used for his experiments has been lost in the Arklay incident, though we aim to determine this through further research. As the organism continued to grow in size, its roots spread deep through wood, concrete, and soil, latching itself onto the aqua ring far beneath the residence. There it fed on chemically enriched water and accelerated into monstrous proportions. Vines erupted through cracks in the dormitory floorboards, twisting across walls and ceilings, and entire rooms became its hunting ground. Its body rooted in place, but its reach extending far beyond the original point of infection. Unlike normal flora, Plant 42 exhibited behavior that blurred the line between plant and predator. Its vines could manipulate objects, even turning doorknobs to ensnare prey. When Umbrella staff attempted to destroy it, the plant in turn responded with aggression, both striking, constricting, and draining its victims of blood. Its physiology is unique, wherein large ivies act as both sensory and offensive organs, capable of whipping victims with lethal force. But not only that, specialized sacs developed within its body allows it to eject toxic fluids to weaken targets before feeding on them. Its vascular system, enhanced by the T-virus, has adapted to absorb blood directly, using it as a supplemental nutrition besides the use of water. In this way, Plant 42 did not simply use photosynthesis, it was also a consumer of organic material. Our research indicates that by late May in 1998, the plant had completely overtaken sections of the dormitory. Several attempts by the researchers to neutralize it failed, whether this was due to the infection of the staff or interference from other biohazards within the facility is unknown, yet Plant 42 remained unchecked until the arrival of stars in July that very same year. From a tactical standpoint, Plan 42 is a paradox for us, for just like Yawn and Neptune, it is immobile, yet lethally effective within its territory, and despite being vulnerable, it demonstrates yet another fascinating ability within the T-virus to grant predatory behavior to organisms we never even intended to use as weapons, and though it is a weapon that cannot be moved or deployed, I highly suggest, Director, that we look further into this project, as it is a reminder that the T-virus obeys no boundaries. This is Specimen 011, also known as the Black Tiger. This arachnid was the result of prolonged and unstable T-virus mutation within the web spinner strain. While most infected spiders reached a size of several meters, this particular specimen exceeded them by far, growing up to 3 or 6 times their body mass. The black tiger is distinguished by its black and yellow fur, and became the apex of its kind, a grotesque matriarch ruling the caverns beneath the Arklay facility. The silk of the black tiger was immensely more thick and resilient than that of its kin, strong enough to coat entire tunnels and chambers, trapping intruders as prey. Within the underground caverns, its web became labyrinthine snares, each thread a silent alarm to any movement. Its physiology mirrored and amplified the traits of its lesser kin. Enlarged venom glands allowed it to spit toxin across several meters, a corrosive fluid capable of weakening or even killing its victim before the creature would then close the distance. Its fangs were long enough to puncture bone, delivering venom directly into the bloodstream. And we have documented reports that show survivors going into paralysis and violent convulsion within minutes of contact with the venom. Behaviorally, the black tiger exhibited maternal instincts corrupted by the infection. It became the mother of a cluster of smaller arachnids, all of them protecting its nest aggressively. These offspring we have yet to name were less developed than the full web spinners, but added to the danger of the black tiger, as they would swarm any victim while the black tiger attacked with overwhelming force. From a tactical perspective, the black tiger was more than an oversized predator. Its ability to control an area with webs, its lethal venom, and its resilience to small arms fire made it one of the deadliest organisms encountered in the Arklay incident. Its presence also confirmed the instability of our arachnid projects, and though we have deemed the web spinners to be too unpredictable for further development, and seeing that the black tiger emerged as a consequence of neglect, we ultimately view the black tiger as a dead end as well, a simple byproduct of the T-virus left unchecked. And though we appreciate the promise within the project, we simply deemed there is too much risk and not enough reward in pursuing projects that are saturated with instability. Finally, we arrive at Specimen 012, the Tyrant Model T-002. Unlike the zombies, the Cerberus, or even the Hunter Alpha, none of this creation was a part of an unintended consequence. The Tyrant was the pinnacle of the Arklay Laboratories research, and it is the culmination of the Tyrant project, this particular branch's best attempt at creating the ultimate biological soldier, intelligent, obedient, and unrelenting. For years, the progress of this project was stalled by one obstacle: genetics. For as it turns out, our researchers have uncovered that only a fraction of the human population carry the rare gene that allows the T-virus to produce a stable mutation, rather than the degeneration and instability that we see in practically every other specimen within the Arklay file. Even when such candidates were found, the mortality rate during infection was catastrophic. Most subjects perished before transformation could even be completed, but the T-002 represents the first stable success. Therefore, the Tyrant was not left to mutate naturally. Its transformations has been guided by surgery and genetic manipulation. Most notably is that its cardiovascular system has been entirely replaced. Surgeons transplanted hearts from 2 large mammals, altering them to function as a single dual system organ. This physical enhancement allowed the Tyrant to survive massive trauma while maintaining a blood flow required to fuel its monstrous physiology. The final result of the project was a giant of a man, nearly 3 meters tall, its flesh thickened and reinforced, and its musculature pushed far beyond that of regular human limits. Its left arm developed a grotesque weapon, bony, elongated claws capable of tearing directly through steel. Yet as previously mentioned, unlike the zombies, the Cerberus, or essentially any other specimen within the Arklay Files, the Tyrant's body showed no sign of rot or decay. The T-002 was alive, stable, and evolving. Neurologically, the Tyrant retained more function than our researchers expected, and while brain damage did occur during infection, it remained capable of higher reasoning, showing awareness and hunting tactics far beyond that of the shambling dead. It was intelligent enough to follow orders had our researchers chosen to release it as planned. According to the logs, the T-002 was intended to be tested against combat veterans in live fire exercises, after which it could be cloned for mass deployment, yet that deployment never came, for ultimately it was not released for research. But what our current investigation suggests is that one of our own researchers, Dr. Albert Wesker, in a rogue campaign against the STARS unit, unlocked the chamber and unleashed the Tyrant during the Arklay facility's collapse, hoping that it would wipe out his own team. Instead, the creature turned on him, killing him in the process, before it then hunted the remaining survivors within the ruins of the laboratory. The Tyrant's final transformation occurred when its limiter was destroyed. Without the limiter suppressing the T-virus within its body, it entered a second stage of mutation. Its muscles thickened further, its skin flushed with blood, and its speed and power multiplied to superhuman levels. At this point, we have dubbed it the Super Tyrant, a creature now capable of smashing through reinforced concrete in its pursuit of its victims on the surface. Video footage from the helipad suggests that the Tyrant was destroyed by a direct strike from a rocket launcher, and that the biological remains of the creature was incinerated in the Arklay facility's destruction. And though we consider the physical loss of the T-002 a catastrophic detriment, all hope is not lost, Director, for all logs and research entailing the Tyrant project has been recovered, and we deemed this project and this creature is but a small glimpse and a giant stepping stone in terms of improving the future projects and research which we intend to undertake. Everything within this report has already been forwarded to headquarters, yet if you should have any questions, I am here at your disposal. Hey there, Hazmat here. That concludes one of my more ambitious projects, and if you've enjoyed this episode and would like to see more in this particular style, then make sure to let me know in the comments below. As always, a big thanks goes out to the Hunters Lounge, specifically the exclusive Johnny Sevens, the exclusive Beach Boy, the exclusive War Monk, the exclusive The Cayute, and the exclusive Rat. The VIPs, Volvo Bear Slayer, Derivative, Doc Powell, Gino Lucero, Goose, Hambone, Jimenel Bob, Corey Peterson, Michael Furnell, Mordef Menace, Mr. Mouse, Ryan, Hein, Slip, Stas Piestek, Sunrise Phoenix, TJ Crane, Texas Toast, and the Wizard of Copper. And of course, all the regulars. If you like the content I produce and would like to support the channel, consider signing up as a member on the Patreon for as little as $1 a month. The link is in the description below. And thank you all for watching. I'll see you in the next one.